Okay. Okay. Yeah, so sorry for the late start today, but I think we're okay now. If any of you are having trouble seeing this or hearing me, well, then you wouldn't know how to respond, right? So anyhow, let's continue our discussion about cell proliferation and how our cells maintain or regulate the number of times that they go ahead and duplicate or replicate before they stop. So this has to do with telomeres and telomerase. So I think most of you probably are, at least in passing, familiar with what the telomeres are. And we're going to explore that a little bit more in depth today and talk about why the maintenance of telomeres is so important, not only for the normal physiology of a living cell, but also how mismanagement of the telomeres can be a big contributing factor towards the development of cancer. So let's review some of the key concepts that we've been talking about. We said that with every division that a cell makes, the daughter cells will gain wear and tear on the genome. Remember, we had used that example of the genome being a photocopy of a photocopy every time that a cell divides. Right? And even though you have stem cells that persist in your body as multipotent stem cells, right, they are accruing certain damage, wear and tear, right, over the many years of your life. And I wanted you to consider that every time you have a stem cell that gets some kind of damage to it, right, any additional daughter cell that it gives rise to repopulate tissues will carry that additional bit of damage as baggage. Right? And that's something that I think we don't really consider, because we just think about our stem cells as always just kind of replenishing the healthy differentiated cells of our body. But of course, if you stop and think about it, right, whatever damage is in the genome of those stem cells, that becomes the new version of the genome that gets put into all those differentiated cells. That's why last time we looked at an example of why the skin of a teenager, right, is so different than the skin of a senior citizen. And I asked you to think about, we all know that old people have fragile skin, but why is that, right? If in reality they have stem cells that replenish it, it's because stem cells lose their proliferative potential over decades of life because of little bits and pieces of damage to the genome. And depending on what those damages are, they are getting passed on to result in keratinocytes in the skin that are not as good by the time you're 80 years old as the keratinocytes in your skin when you were 18 years old. Right? So with every division, we say daughter cells gain wear and tear. The cells must know when they should not divide further. Right? So evolutionarily, there must be a mechanism so that those additional damages are not persistent and passed on, right, to more and more cells. So that's why differentiated cells may only proliferate like 4 or 5 times, right, before they senesce. Right? And senescence, we said, is triggered by increasing levels of CDK inhibitors. Right? So we looked at last time how once a fully differentiated cell, right, has divided a few times, it has upregulated certain CDK inhibitors like P21, and that's one way it goes ahead and goes into senescence and stops. Right? And that's how they evolved, to protect you, to protect your whole body from them going too far. So let's think about this idea, of baggage, right? So we said you have an original stem cell somewhere. This could be one of the multipotent stem cells in your body right now. And as its job is mainly concerned with proliferating to replace all the other fully differentiated cells that are being lost all the time, right? You have the starting genome, right? This is what it's supposed to be. And let's say you have somewhere in there a gene, right, on one of the chromosomes that is transcribed, right? And this is the RNA that comes off of that gene after it's edited, and then it's put together as mRNA, which is then translated, right? And then a certain series or sequence of amino acids are then assembled as the resulting polypeptide chain of that protein, right? So remember from your cell biology class, you have the start, the translation start right here. So this would be methionine, right? And at some point, you would have a stop codon, right? And that's where the polypeptide chain ends. So when that cell divides, it makes two daughter cells. And so we can say, let's pretend that the message here, right, encoded in this amino acid sequence is DNA has all you can ask for, right? So that's the normal sort of resulting message represented by these amino acids in terms of their proper sequence and the proper folding of the protein. Right? But let's say one of the daughter cells, right, takes a hit. So then the daughter cells that it gets rise to, so let's say you get a silent mutation, right? So the U over here gets replaced by an O. Well, it still kind of works because it still is DNA has all you can ask for, right? So maybe the reality is it's kind of neither here nor there, right? But the point is that becomes part of the genome, right? As this lineage over here keeps dividing, then you might get different types of damage. For example, a missense mutation, right? Or a nonsense mutation. Right? And eventually, right, over many, many generations, we're not really just talking about 1, 2, 3 divisions. Remember, we learned last time, right? It takes many, many, many subsequent divisions for something like a transforming cell to really build up all the random baggage of mutations that actually bias it towards being, you know, a cancer cell. So eventually you get to the point, right, where something like a frame shift happens, and now you've really lost the meaning here, right? So by then, too much information loss and risk of cancer. Right? So at this point, this is when the cell should undergo apoptosis, right? So P53 should be able to detect, you know, there's a big problem here, right? And if we cannot fix this, then just forget it, right? So that's why this red cell over here should take itself out of the picture, right? So that you really just have these nice healthy cells instead. Right? And so cancer happens, right, if for some reason apoptosis doesn't take place and the cell increasingly gets more mutations, and those mutations may bias it towards more and more rapid proliferation. So in order for cells to become cancer cells, we said they have to become immortal, right? Because normal cells, even cells that are, you know, transformed a little bit on purpose to make cell lines, right, that last a lot longer in the lab, those are still not truly immortal, right? Because we said even the cell lines that you use in the lab, you passage them too many times and they're still going to stop, right? So at that point, you throw them away, you go back to your frozen stocks of your cell line and you start like another culture, right? So you have a fresh batch of cells, right? But cancer, of course, keeps going, right? So in order to achieve that kind of immortality, cancer cells face two hurdles. Number one is overcoming senescence, right? That's what we've been talking about. So hopefully, at least intuitively, that makes sense to you. And number two, they have to overcome an event that's known as crisis. Right? So here, you know, these cells have transformed to the point where they are showing various stages of things like senescence, crisis, and apoptosis. Right. So I should say these are not necessarily transformed cells. These are just cells that have gotten to the point where many of them are being subjected to them having to stop their proliferation. Right. So it's all about this idea of immortality. So I think hopefully you guys know who this is up here. In past semesters I've asked, and students are like, that's Han Solo, right? I'm like, okay, yeah, close enough, right? Anyhow, so let's remember this. Each chromosome really is just one single long molecule of DNA, right? So even though the human genome is 3.2 billion base pairs of DNA, it's divided up into several long individual DNA molecules, right? Each one of those is one of our chromosomes. And we know, right, that the chromosomes are not just naked DNA floating around. I think all of you guys know by now, right? DNA is packaged up with different types of proteins to form chromatin. But that doesn't change the fact that it still is, one chromosome is one long piece of DNA. So that means you're going to have two free ends of the DNA. And why is having two free ends of DNA a problem? Well, the free ends might be more easily damaged because they're just kind of like hanging out there. Or, right, the free ends, if you have different DNA strands floating around, if they just have free ends, the free ends of DNA strands might stick together. That could really be a problem, right? If you imagine two chromosomes just attaching to become one large chromosome, that's not good, right? So ends of your chromosomes need protection. I think of it kind of like your shoelaces, right? Your shoelaces, especially like your sneakers or something, wear them a lot, the ends get all frayed and kind of ratty. And so you have these, these little things at the end of your shoelaces, which, you can just take a piece of Scotch tape and do it. But a brand new pair of shoelaces, right? They already have these little plastic things in there. I think it's funny because that actually has a name, right? I think those things are actually called like aglets or something like that. I think that's what they're called, right? One of these random little Trivial Pursuit things. So you have to protect it, just like you can protect the end of your shoelace by having a little bit of tape wrapped around it, right? So does the ends of our chromosomes have certain types of ways of being protected. Right? So that's what the telomeres are. So telomeres are DNA, DNA that's associated with certain proteins to make like these little aglet-like things, right? The little Scotch tape that wraps the ends of the shoelaces. But what's really going on in there? So telomeres are tandem repeats of TTAGGG over and over and over and over and over again at the ends of your chromosome. So if we were to imagine the middle of your chromosome has all kinds of very important, very valuable DNA sequences that represent your genes, right? If you keep going towards the end of the chromosome at a certain point, right? So you'd have most of it being non-telomeric DNA. So you'd have many, many megabases of still different sequences, things like that. Then once you get closer and closer to the end, you no longer have important genes there anymore, right? You start having sort of just these repeated telomeric sequences, right? So you have TTAGGG over and over and over again, right? So a good 5 to 10,000 base pairs of these telomeric repeats. Right? Notice also that the double-stranded DNA at the end, it doesn't just end very cleanly. The telomeres have this overhang, a 3' overhang, where the pink strand in this picture, the G-rich strand, because that's where all the G's are, right, overhangs the C-rich strand. And that overhang is about several hundred bases long. So why is this important? I often think about, when I was a little kid and I'd look at a book, right, I always wondered why when you open the cover up and maybe there's like a title page, there's always some extra blank pieces of paper in there before you actually get to, let's say, a table of contents or something like that, right? And I always wonder, wow, what a waste of paper. Why do they bother with that, right? But if you think about it this way, right, those extra pieces of paper are kind of like protection, right? Because if those extra pieces of paper that are more exposed because they're on either side of the book, if those get torn out, it's okay, right? If those get ripped and torn out, you're not losing any valuable information. You're trying to protect the text part of the book that has the important information, right? And so therefore, telomeres are like that as well. They are like the expendable free blank pages in a book. All right, they can be lost without actually you losing important genetic information that's part of the chromosome. So telomeres not only protect the ends, because free ends of nucleic acids, I may have mentioned this to you before, but our cell, right, is never very happy having just free-floating nucleic acids in it, because evolutionarily, right, free-floating nucleic acids, especially double-stranded nucleic acids like DNA or RNA, that looks a lot like something that might have gotten introduced into the cell by viral infection, right? So some sort of pathogenic infection. And so as a result, our cells have evolved to not like the presence of free-floating double-stranded nucleic acids, and we have certain enzymes that just are basically in there in our cell chewing up to destroy potential viral DNA. Okay. That's also why when, another reason why, of course, our DNA is protected by being held tightly in the nucleus, right? So it's not subjected to as many of these types of patrolling enzymes out in the cytoplasm that are looking for, suspect DNA or RNA to chew up. And that's why when the mRNA is edited from being transcribed from a gene, right, and then edited to get the mRNA that then needs to be shipped out of the nucleus into the cytoplasm in order for ribosomes to be able to do the translation. That's why mRNA, right? I mean, number one, its mRNA is single-stranded, right? So it doesn't look like DNA to start with. But number two, I think you all know, the mRNA has different things on its end, right? Like a 5' cap on one end and a 3' poly-A polyadenylation tail that serve not only as signals for different proteins to bind the mRNA and transport it out of the nucleus, but also they protect the RNA from being chewed up by things like these exonucleases, right? So remember, exonucleases are enzymes that recognize free ends of DNA and chew from each end coming in. That's different than endonucleases. Endonucleases can bind, right, to DNA somewhere in there and just cut at different spots. So that's why if you're familiar with doing labs and you do restriction enzyme digests, right, using things like EcoR1 or Hindi3 or BAMH1, what are those, right? Those are endonucleases that come from bacteria. Right? We don't make those types of endonucleases in our cells, right? Because that wouldn't be good if those things got into our nucleus and they would just start chewing up DNA. But in bacteria, they make those enzymes so that phage or viral genomes that are infecting the bacteria, right, can be chewed up and digested. Okay, anyhow, so having telomeres protects our DNA from exonuclease activity. Also, they protect the ends of our chromosomes from sticking together improperly. And importantly, they provide a much needed physical platform in order for DNA replication to happen. Let's look at number 2 here, prevents chromosome sticking end to end. So in this figure, this is what a normal sort of karyotype would look like, right, of a healthy cell, normal chromosomes. So you'd have 46 chromosomes here, right? So you'd have 22 pairs of autosomes and then a pair of sex chromosomes mixed in here somewhere. What they've done here is they have in red, they're staining DNA, right? And in green, they have some sort of a marker that stains certain telomeric proteins that are part of these telomere complexes that protect the ends. So this is neat, right? You can see the little green dots are at the ends of the chromosomes. So those telomeric proteins are there. So these are normal, healthy chromosomes that have those caps on them to protect them and prevent them from sticking together. How important is that? Well, look at this, right? So if you have a defect in production of normal levels of things like these telomere maintenance proteins, right, you might end up with something like this. This is a jumbled mess of chromosomes that are all sticking end to end because they don't have proper telomeres, right? That is really scary, right? And so obviously, you wouldn't imagine that if something starts going wrong in your cells, all of a sudden it would look like this, right? But what if in reality you had a chromosome that due to some sort of weird translocation or something like that, right, it sort of messes up one of its ends and now it doesn't have a telomere on it, right? What if that happens to another chromosome, right? Oh boy, and what if those two chromosomes come together? So telomere loss, right, and segregation issues can happen during mitosis. During mitosis, in a normal scenario, you have your duplicated chromosome here. So the two sister chromatids are still joined together, right? And now during mitosis, each one is going to be associated with spindle fibers that are attaching to the two centrosomes. And then they're going to get pulled apart, right? One to the left, one to the right, one each into the new daughter cell. Since these things are paired up together, right, this is certainly a scenario where the ends of two molecules of DNA are really close to each other. So fortunately, you have these little red telomeres on the ends to prevent them from sticking together, and they can separate properly. So this is what I was talking about a moment ago. What if, for example, the telomeres are eroded away, right? Either because of lack of proper expression of telomere maintenance proteins or for some other reason, right? Some weird translocation break or something like that. Or, as we will talk about in a moment, normal wear and tear. Then you might have unprotected chromatid ends, and then they might be able to stick together. Right? As a result, they will fuse. And where they fuse here, if you think about this, right? If you were to read the nucleotide or base sequence from the left leading across this point of fusion, right? Would there be anything that really stands out that lets you know, oh, this is some weird spot where the DNA joined together improperly? No, right? Because it would just look like one continuous strand of DNA. So then what happens if you have these fused chromatids during mitosis that then start getting pulled apart? Right? There's no reason to believe that that spot where they stuck together is any more weak than any other spot around this whole sort of blue loop here, right, of the two chromatids together. So as a result, right, as they're being separated, look, they will just break somewhere else, right? So here's where they fuse together, but now as they're being yanked apart by the spindle fibers during anaphase, right, look what happens, right? So you get this weird thing now where obviously now the genetic material is messed up, right? You have one version of it that lost a whole bunch of genetic material, and the other sister chromatid now has additional genetic material. So if this is happening on one chromosome in the cell, right, this blue one, hey, it's probably happening on other chromosomes as well, like this brown one, right? So this brown one now has this kind of sticky end, right? And look, it goes ahead and it joins up with the blue one. And now the next round of mitosis, right? Things just get worse and worse. These two now get yanked apart and you have a new breakage here, right? And so now this chromosome, we'll pretend it's supposed to be brown colored, right? Suddenly at the end, there's a bit of blue, right? So we call this phenomenon breakage fusion bridges, or BFB. And hopefully from this explanation, you could see how this could happen, right? If this is not detected by the cell, and the cell does not do the right thing by accepting the fact that something like this can't be fixed. Because I mean, look, how is the cell going to fix this, right? Because it doesn't know the difference between brown DNA and blue DNA. It's just all DNA, right? How would the cell fix this? So a cell that detects this should undergo apoptosis, right? And not allow this to happen for another round of cell division. But what if the proteins that are involved in detecting this, right, are themselves somehow compromised, right? Because after all, this cell would depend upon different proteins to properly see this is happening, right, and initiate apoptosis. And the whole process of initiating apoptosis involves many proteins working together. If any of those proteins, right, are now defective and apoptosis can't happen, this process of BFB will continue. Right? So the genomes of subsequent daughter cells are increasingly damaged, and hopefully the signals for apoptosis are initiated and the cell is destroyed, right? But if it doesn't undergo apoptosis and this keeps going on, right, that's how we get back to this. Right? Remember this, I showed you this at the very beginning of the semester, right? And I'm sure many of you, if not most of you, were kind of marveling at the fact that, like, how does this even happen, right? How could this happen, right? Now you know, right? So if you have a scenario where things are going really wrong inside of the cell, Right? Telomeres have maybe eroded away or somehow are not maintained. And once these BFB bridges, breakage fusion bridges, start happening, if for some reason other genes that code for apoptosis initiating factors, if those also are mutated and compromised, right, eventually over time, you get this type of crazy karyotype, right? The crazy genome of an aggressive advanced cancer cell. All right. So telomeres, I was telling you how you can think of them as the little aglets or whatever, or the little scotch tape that you wrap around the end of your shoelaces. But the reality is, it's a little bit more complicated than that, right? The shoelaces, if you think about it, one way you can protect the ends of shoelaces if you don't have scotch tape is you can kind of loop them around, right, and tie them to themselves. That way there is no free end, right? It's just a loop. And that loop, of course, is not going to fray as easily compared to if you just had a naked end of the threads sticking out. So just like shoelaces can be tied around into sort of a loop to keep from fraying, that's actually what our telomeres look like structurally. So you can see here in this illustration, once again, right, you're approaching the end of a chromosome. And as you get to the end, you're going to have these repeats, the telomeric double-stranded DNA. You have the blue C-rich strand, and then the opposite pink G-rich strand, right? And they loop around. And you can see now why. Remember, I told you that the G-rich strand, there is an overhang, right? That overhang is what facilitates this loop, because you can see this overhang actually ends up tucking back in. So this section of the double helix has separated so that the pink looped around part can actually bind to, right, or anneal to the blue part here. It's all repeats of the same telomeric sequence, right? So it's designed that way so that it can bind in there. And that then loops it around, and you no longer have a frayed or potentially frayed end of the DNA, right? So here's another illustration of that. Looping around, here's the 3' overhang, and it gets tucked back in here, right? Here you can see on an electron micrograph image, this is the telomere, right? It loops around, and somewhere in here, right, the overhanging strand is just tucked back in there. And of course, surrounding all of this, this is just naked DNA in the image here. But in reality, right, this whole thing, you might not even be able to see it because the DNA would be covered with special telomeric proteins, right? So it'd be more like this, right? So you'd have all kinds. I mean, I'm not going to go into detail about what the identity of these different telomeric proteins are, because to me, that's not important for our discussion, right? What is important is that they coat and maintain this proper telomeric loop structure at the end. Right. So we said, okay, that's how DNA ends are protected by telomeres, so that exonuclease enzymes don't just see like a naked double-stranded end that they can chew away. The telomeres also protect the chromosomes from sticking end to end, right? And the third important thing is they provide platforms for DNA replication. So remember, when DNA is being replicated, for example, you have a cell that's been pushed over the restriction point and is leaving G1 into S phase, right? And during S phase is when the genome needs to be replicated. Do you remember our chromosomes? They begin to replicate by having all along the lengths of the double-stranded DNA, these little bubbles forming, right, where they separate. And these are these origins of replication. And then on one side, right, you have a leading strand of new DNA. So the DNA polymerase can read very easily in one direction this way, laying down a leading strand. But right, the other strand, the other template strand, has to be copied going sort of backwards, piece by piece, through these Okazaki fragments, right? Because the DNA polymerase that makes the new copy can only do so in one direction. Right? So that's why you rely upon these little RNA primers. RNA primers are laid down at sort of random intervals so that the DNA polymerase can go ahead and, in the pink here, fill in, right, the new strand, and then the RNA primers are removed, and then little gaps are filled in, right? So as the DNA is unwinding, so let's imagine, right, that this bubble, this origin of replication, is taking place really close to the end of the chromosome, right, really close to the telomere here. So as you're unwinding this, right, these random little green RNA primers will have an opportunity to bind there, right, like this, okay? But think about this. This blue strand represents the very end of the DNA molecule, right? The last telomeric repeat. What are the chances that an RNA primer would perfectly be put right at the end so that it actually is able, right, to capture or still take into account these last few bases here? It's very unlikely, right? In fact, it's more likely that the last RNA primer would be one of these somewhere. So what happens to this bit of the telomere in the original template DNA? Right? You don't copy it. Right? So the new strand of DNA is missing a little piece of the end. Okay? Therefore, with each round of replication, our chromosomes are actually getting shorter because you are actually losing those free end pages of the book. And let's say you have 3 free end pages, right, on either side of the book, right? You're beginning to lose those. At a certain point, what? You risk losing now the actual text of the book, right? And in this case, as your chromosomes are getting shorter with DNA replication, at a certain point, you risk getting into the actual coding part, the important part of the chromosome that contains information that you cannot afford to lose. Right? So that's what crisis is. Crisis is when, after repeated replication of the cell and repeated replication of the genome every time, right, the telomeres have gotten worn away to the point where now it's kind of dangerous, right? Like the next time you try to replicate this and you lose a little bit more of the chromosome, maybe you'll start losing important genetic information. So with each replication, chromosomes lose anywhere from 100 to 500 base pairs of DNA. Once the total telomere length on one end is down to around 100 base pairs, that's when evolutionarily, right, our cell says, whoa, okay, time to stop, right? Replication should not be allowed to continue because now there's the chance that you're getting into the blue part, the body of the chromosome. So let's look at this. Here we have an interesting experiment done in the lab where they had a cell line that they are passaging, right, or dividing. And so each sort of column here represents another round of replication of taking these cells, seeding them on a dish, right, encouraging them to divide again, generation after generation. We said that a normal differentiated cell in your body might only be able to do this 4 or 5 times because, right, a normal cell in your body, it's very conservative, right? But a cell line in the lab, you can really force to go longer and longer. And what they've looked at here on the y-axis is the telomere length, right? And so as they run this out on, I think this is like a southern blot or something. So they're just looking at DNA. This is the telomeric DNA that they're looking at. So maybe it starts off around like 8 to 12 kb worth of telomeres, right? But look, with each cell division, the average telomere length, right, in the next generation of cells that they then harvest and lyse and take the DNA out and run it on the gel gets shorter and shorter until you start getting down into this range of closer and closer to, let's say, 1,000 base pairs, right? And once you start getting sub 1,000, right, then you really start getting into problems. Because at this point, you can't even tell because the average base pair size is so small, you can't even tell where it is anymore, right? So this would be when crisis takes place. And a normal, well-behaved cell, right, this is where apoptosis would put an end to it, okay? So in a cell line, it may be that they just stop dividing and they just sit there because they senesce, right? But if you have a cell in your body that's supposed to be actively dividing and not senescing, maybe something like a stem cell right? Then if you really are encountering this type of problem in the stem cell, then it should undergo apoptosis. What about cancer? Right? This actually turns out to be a cancer cell line that I'm showing you, right? So in this case, what it's showing you is that as you keep pushing it beyond crisis, the cells don't die, right? Somehow they are maintaining enough telomere length in them that they can keep going, right? What do I mean by they're maintaining enough telomere? Right? Because I thought we just said that every time DNA replicates, you lose a little bit of it, right? Well, clearly, right, the cancer cells need to have intact telomeres, because if the cancer cells didn't have intact telomeres anymore, then all the chromosomes would just stick together, like that super messy picture I showed you at the beginning of the lecture, right? Or just look like a big pile of red spaghetti, right? A cancer cell cannot survive like that, because after all, a cancer cell is still a living cell, right? A cancer cell still needs to have a genome that it can rely on to make all the different proteins that keep it alive, right? Once you have that big stuck together pile of red spaghetti, right, that's not going to work, right? So somehow cancer cells are maintaining a nominal, sort of minimally acceptable length of telomere. And that's why you see the smear on the slide here. It doesn't get any shorter and shorter, right? It hangs out around, let's say, 1 to 2,000 base pairs. So how does it do that? How does it do that? They rejuvenate telomeres using an enzyme complex known as telomerase. So telomerase is a big protein complex. In our bodies, we have what we call hTERT, right? So it's coded for by different telomerase genes that make the different subunits, protein subunits of this big hTERT complex. And the way hTERT works is it's really kind of interesting. See this blue strand here, right? This is a strand of RNA. It actually is a free piece of RNA that's specially coded for by our genome that represents the template to sort of stamp out more telomeres, right? So it is the template of the telomere sequence, the TTA, GGG, TTAG, right? And the hTERT uses that RNA template and hangs onto it, and it adds, right? It adds to the end of the telomere more telomeric repeats. Okay, so that's why maybe the telomere started at this point here, but after hTERT carrying around this HTR RNA template, added more TTA GGG sequences, you are now extending the telomeres again, right? So, this hTERT essentially is doing what? It's doing reverse transcription, right? Starting from RNA and reverse transcribing the RNA to make DNA. So what does this ability to encode DNA through reverse transcription of RNA remind you of? What about retroviruses, right? Retroviruses have reverse transcriptase, right? So that RNA genomes of the viruses can be reverse transcribed into DNA. Is there connection here? Yes, right? Because remember, one of the cool things that we learned at the beginning of the semester is how do many of these tumor-causing viruses actually get things like viral oncogenes in the first place, right? They stole them from the host genome. Right? So I don't know if it surprises you, but the reverse transcriptase itself that's used by RNA viruses, retroviruses, at some point was stolen, right, from the host genome, and it mutated in such a way to become the retroviral reverse transcriptase. So if you look at this here, P123 is the name of the human TERT telomerase complex, right? So it has these different sort of gene components here, color-coded here. Below it, right, are the reverse transcriptase genes found in different reverse transcriptase retroviruses, right? And you can very clearly see, it's so obvious, right? At some point in viral evolution, right, these were the genes that accidentally got copied into the virus, right, and became usable by the virus so that it could persist and have an RNA genome that would be reverse transcribed into the newly infected host cell, right? And this mechanism turned out to be so important that that's why you have such diversity now of these types of retroviruses, right, that infect us, right? But some point, I'm not even talking about millions of years ago. We're talking about like hundreds of millions of years ago when you first started having eukaryotic cells, right? This event took place, right? So this is the, for example, this is the reverse transcriptase in HIV, right? So this is what enables HIV to infect cells. And this is the reverse transcriptase protein, and they've color-coded it to show you, right? Indeed, right? Each one can map back to the normal human version of TERT, right? I think this is like shocking when you realize this, but then hopefully in your mind, you're like, wow, yeah, of course, right? This is how it happened. Okay, so viruses borrowed our genes and turned them against us, just like borrowing C-Sark and turning it into V-Sark. So here's an important concept. hTERT expression in our cells is normally high in stem cells and pluripotent cells. That's how stem cells last, potentially, right, for decades, because they're dividing. Every time they divide to replenish additional cells, aren't their telomeres getting shorter and shorter? Yes, right? But in stem cells, they have the hTERT genes activated so that they make the hTERT complex, so that they're constantly, as telomeres are shrinking, right, they can lengthen them again so that the stem cell can live another day and go for another round, right, of proliferation for decades and decades. In our fully differentiated cells, like your skin cell, muscle cell, whatever, of course, it's not like they've lost the hTERT genes, because every nucleated cell in our body has the same set of genes. It's just in those cells, right, the hTERT locus has been shut down, right, shut off pretty much permanently, so that differentiated cells no longer make hTERT, and therefore their shortening of telomeres becomes a very useful sort of keeping track or a tally of how many times they divide, right? A fully differentiated cell will maybe only go for 4 or 5 times, and then they hit crisis, and then they undergo apoptosis, or at least they enter senescence, right? So that they no longer do anything and no longer pose a threat of dividing anymore until they are recycled and eliminated. So this last sentence here is really important. The hTERT gene is still part of our genome in all of our differentiated cells, right? And so what if the hTERT gets reactivated, right? So this is now what we see, right? So if you look at hTERT re-expression in tumors, this is what you see, right? These are Kaplan-Meier plots, and if you look at patients who at the time they were diagnosed with a certain type of cancer, the pathologists in the lab decide to look at hTERT levels, whether hTERT can be detected as being woken up again in these tumors at the time they were first biopsied and diagnosed, right? And then they break them into 2 groups. One cohort are those who have tumors that when they were first presented in the clinic doesn't look like there's evidence of hTERT being woken up again, right? And there's the other cohort. From the biopsies, the pathologists can tell, ooh, right, there's a lot of hTERT protein in these cells again, right? And look at the dramatic difference. This is neuroblastoma. So years since diagnosis, right? So depending upon if that tumor had telomerase reactivated or not, it makes a big difference on patient survival, right? Same thing here with Ewing sarcoma, a different type of cancer. Right? Low telomerase versus high telomerase in the patient's tumors. So this is the last slide. I'm sorry we're going over because I had a hard time starting today. But bypassing senescence and crisis, right, is what transforming cells have to achieve in order to become cancer, right? Because they need to achieve immortality. Right? They have to get around the roadblocks of undergoing senescence, right? They have to get around somehow the challenge of shrinking telomeres that are serving as their normal tally or clock, right? To keep track of how many divisions they should do. So let's look at this scenario here. Here we have the end of a telomere. If the telomeres get too short, right, chewed away from replication or whatever, so then you have, right, the telomeres are starting to get too short. There's no overhang anymore, and there's DNA damage. There will be certain enzymes, right, proteins that detect these, like the checkpoint enzymes, that will then phosphorylate P53. Right? So if you go back and look at those cartoons that we looked at about P53, normally, right, it is ubiquitinated and then degraded. But if it doesn't get ubiquitinated, right, and it gets phosphorylated in certain spots to help it escape the MDM2 that wants to degrade it, then the P53 will activate, right? And remember, the things that phosphorylate the P53 were ultimately proteins like checkpoint kinases that link back to detecting something wrong with the DNA, like this, right? So activating P53 would then cause this cell to do the right thing and go into senescence, right? But what if the cell has improperly reactivated telomerase, right? That means the arrow goes back in this direction, where you keep having the telomeres being maintained, right? So therefore, this cell would not undergo senescence or crisis, right? Consider also that SV40 large T antigen, right? Certain tumor viruses that prevent P53 from working or prevent RB from working, right, can also be a way to avoid senescence, right? So as we will explore this further, we'll look at how certain types of mutations, right, happen in certain orders, typically, as a cell is transforming, in order for sort of like the right series of events to happen that allow the cell to really achieve immortality like this. Okay. All right. So that's the end of our lecture.